Hi everyone, my name is Michael, and I'm a master's student in human genetics at McGill University in Hamed Nashrafabadi's lab. I'm here today to present you my poster, MPAQT, a novel data integration framework for isoform quantification with short-read and long-read RNA-seq. Quantifying transcripts using only short reads is a challenge because it's hard to know with certainty which transcript that read comes from. So as a result, similar transcripts get misquantified. And these small differences in transcripts can have important biological consequences. We've developed MPAQT, which uses a generative model to connect transcript abundance profile of a sample to the expected long read and short read distributions. And this allows us to get inferences of transcript abundances using only short read data or combining short read and long read data. We've benchmarked MPAQT using real and simulated data sets, and we show improved performance at both gene and transcript levels with and without long read data. Our quantification framework uses the concept of an equivalence class, which is a set of transcripts which produce equivalent reads. Some examples that you see here are equivalence class A and equivalence class AB, and this one is shared between the two transcripts. We have this reference matrix here named P, which we generate by simulation, and we map reads to equivalence classes, giving us the expected number of reads in each equivalence class produced by one unit of each transcript. We then use a generative model to connect the expected equivalence class counts provided in P and the measured equivalence class read counts from our sample to infer transcript abundances. This allows us to perform maximum likelihood estimation of transcript abundances from short read data alone or the combination of short read and long read data. B and C represent two scenarios with differing expected number of reads in each equivalence class in our matrix P, which would result in different quantification inferences. Our first benchmarking is gene-level differential expression benchmarking. We took the log-fold change between two samples and benchmarked against gene-level qPCR values for around 15,000 genes. We see that MPAQT corrects differential expression of genes with low expression that other tools have trouble quantifying. In panel A, we see MPAQT's performance compared to three other tools. These other tools have outliers which are misquantified in differential expression. We see that MPAQT, or that the three tools share about a little over a third of their outliers, and that MPAQT doesn't have any outliers. When looking closer at the outliers, we see that they represent genes with low expression and that the common outliers among the three tools are slightly lower in expression. Next, we benchmarked at the transcript level. We generated simulated samples. We benchmark MPAQT and the three tools against truth TPMs, transcripts per million, which are used as input to a read simulator to generate our simulated samples. We see that MPAQT performs better using two test statistics, two metrics, and across two different types of simulated samples. We then add in simulated long read data to quantify transcripts. We can see that when we add long read counts, MPAQT's performance improves slightly. In panel A, the simulated long read counts are generated with a Poisson distribution. In panel B, we look closer at the differentially quantified transcripts and confirm that they are indeed being quantified better when the long read data is added. We extract these outliers using Mahalanobis distance. In panel C, we see some properties of these outliers. We see that they have more exons, larger gene widths, more isoforms on average, and are enriched for neuronal differentiation genes. We also have real data. We have neuronal differentiation samples where we differentiate human embryonic stem cells into neurons. We have paired long read and short read data for these samples at 3 time points. We take a look at 2 neuronal differentiation genes, CNTN1 and NFE2L2. We see that addition of long read counts allows us to change our quantifications for certain transcripts, which would not be possible with short read data alone. We see that these transcripts differ only slightly at the 5' and 3' UTRs, and it's known that UTR sequences are important for mRNA localization and regulation in neurons. We feel that this differential quantification, this ability to quantify transcripts with long reads, can allow novel research into differential UTR usage and neuronal differentiation and development. In summary, the work we've done is we've developed MPAQT, a statistical framework to integrate short-read and long-read RNA-seq quantification information. We did it to improve quantification of transcripts that are hard to quantify with short-read data alone. And this matters because MPAQT is not only a general-purpose gene and transcript quantification tool, but it can also improve quantification of transcripts with low abundance, complex splicing patterns, and characterize alternative 5' and 3' UTR usage. Thank you for listening to my video, and I hope you all have a great conference.